And I've asked a question. Take it. Yeah.
Adrian's going to
Excellent.
talk about the whale fall side of
Very hard. Very hard act to follow that. Let me see. Okay. Well, thank you very much, Sylvia and Chris, for inviting me, and the Geological Society for hosting me today. So I'm going to be talking to you a little bit today about whale falls, of which you've seen just a few little snippets of information. I'll hopefully give you a bit more of an idea of what on earth we're talking about in this regard. Whales are perhaps the most celebrated, romanticised, discussed, hunted, exploited, regulated and researched of the ocean wonders. And it's astonishing, really, that nobody had ever given much thought to what happens to these things when they die and sink to the seafloor. And this is precisely what the science of whale fall biology is about. It was a really relatively recent story. So the story actually begins in the year 1900, and I'm going to try to explain to you, take you from the story to how this concerns deep sea vents and seeps, which you've been learning about a bit today. But in that year, there was a fisherman off Aberdeen who trawled up a whale skull, a whale bone, and clinging to it were many of these little bivalves, which you've just been learning about from John. And this was very interesting and was published in a small taxonomic paper. And that was the end of the story for a very long time afterwards. Meanwhile, we surveyed the deep ocean. We invented camera systems which would allow us to get to the bottom of the ocean floor and look at the vast expanses of the abyssal plain. We went on in the late 1970s, to discover hydrothermal vents at the Galapagos Rift in the East Pacific. Here I'll just show you a fantastic clip, gratuitously from the National Geographic film Dive to the Edge of Creation, which you can download parts of it on YouTube, and it is a wonderful documentary on very much of its time. It's really amazing. But of course, that was from the second voyage to the Galapagos Rift. The first one, they didn't really have very good video cameras. They just had still footage. When they went back with National Geographic, they put the first proper video cameras on there. And then, almost by way of contrast, moving on in the 1980s, the discovery of cold seeps, such as this. And for those of you who've been learning a little bit about cold seeps today, I recommend to go to the Marum YouTube channel, the Guillemar ROV channel, with the most fantastic high-definition imagery of these remarkable systems. Here are some of those kiwa crabs that John was talking about. So all of these amazing discoveries were taking place, and still really nobody had thought very much about what happens to whales when they die. And let's just think for a moment about whales. What you're seeing here is bubbles of hydrocarbons coming out of the sediment. Have a little think about, these environments are potentially the world's next geofuel. Maybe methane hydrate will fuel the 22nd century. But take back a few hundred years, whales were the first industrial biofuel for many, many hundreds of years. Huge, great ocean liner beasts, hoovering up krill, feeding very, very low, just like the krill fisheries of the Southern Ocean are now, hoovering up krill, feeding very, very efficiently and turning it into oil, and then transporting that oil around on sort of miniature underwater biological oil tankers, and then sinking and dying into the seabed. And that's where the story got interesting, because my colleague in America, Craig Smith, just happened across a whale carcass on the seabed in Southern California and commenced a series of experimental deployments, is one way of phrasing them. Other ways are most bizarre sorts of experiments you've ever likely to get involved with. We've dabbled with whale sinking. It's an interesting experience, put it that way. And this is I didn't play reverse. Got to see a bit of this, even though it's not. Hopefully I've got a mouse. There we are. Even though it's not entirely relevant. This is what happens to a whale when it dies, and of course the flesh gets consumed by these amazing mobile scavengers on the seafloor here, Somniosus pacificus, sleeper shark, and you can see writhing around it, hagfish, Eptotratus, eating the flesh. But from the point of view of you lot and what we're talking about today, that's not the interesting bit. It's very interesting from the point of view of deep sea scavenging ecology, but whatever. Let's think about these bones on the seafloor 18 months to a year or 2 years after that flesh has been scavenged away. You then essentially have a hard substrate structure, maybe as big as this room, with whale vertebra here. This is a vertebra of a grey whale at 1,600 meters water depth. about this big, poking up off the seabed. There are the ribs. 80% of it in certain places is lipids, is oil, and it forms a chemosynthetic community. Here you can see bacterial mat, Vegeatara type chemosynthetic bacterial mat covering the surface of those bones. These are the snowboarding worms that I've been working on that John mentioned. We nicknamed them that because they were kind of surfing down the sides of these bacterial covered bones. Here we are scooping some up with our ROV manipulator arm for later study. And this was a tremendously interesting discovery because it implied that chemosynthesis could occur wherever a whale dies and lands on the seafloor. And this species that we're actually picking up here is a very interesting one because it's also been recorded from hydrothermal vents. And it was these observations right at the beginning. Here he is now, this little polychaete trying to escape, but we captured 12 of them like that, just picking them up individually, brought them back to the surface alive, actually. And but it was very interesting because this led to the theory in the late 1980s in Craig's first paper that these are ecological stepping stones in the deep sea. So if you're a hydrothermal vent or cold seep animal, you could potentially use a whalebone on the seabed as an environment to hop across on your way to the next hydrothermal vent or cold seep. And that was a good theory, and here you can see this actually is the original whale that Craig first found, the Santa Catalina Basin whale fall at 1,200 meters. Here's a vesicomyid, who just learning about from John, sitting next to that whalebone, dipping its foot down and feeding on sulfide that's being emanated from that whalebone. And we think that this whale carcass, based on radioisotope dating, is between 70 and 80 years old. So it's been sitting there for a very long period of time, forming, this is a fin whale or a blue whale, so these are big vertebrae. You know, they're like this big. They take a while to get eaten. But very soon after that, in the late 1990s, with increasing use of phylogenetic analysis, people started thinking about these as being evolutionary stepping stones, and that's what really the focus of what I'm going to try and review for you today. This is a review talk about several papers, not just mine. And so what happened was that they said, well, maybe these whale falls have played a role in the origin and the diversification over evolutionary time. So they're not just important in the sense of animals can hop on and off a whale carcass if they see one, but actually there's so many whale carcasses. Craig Smith has calculated that there may be one at a minimum of every 9 kilometers, sort of nearest neighbor distances on the whale migration routes in the Southern California region, that they're actually pretty important over long evolutionary time periods as well. This was the first genetic study, Amy Bako, looking at vesicomyids, and then that was followed by the paper, which we'll talk about in a little bit more detail now, by Dan Distel, and in the year 2000. And this was the data, and John has showed you these trees, some of these trees, which I'm going to show you again now, but we're going to think about them in terms of their role that they have played in driving evolution at these environments in terms of their habitat use, not just in terms of whether they're chemosynthetic or not. And this paper was entitled Wooden Steps to Deep Sea Vents. And it's amazing. It's amazing how easy it was to get in Nature in 2000, right? That's not to say this paper, it's just a really good paper. It's a really important paper. And but 10 taxa, you know, one short gene fragment. But it was a really important hypothesis. And oh, this is a bit dying, isn't it? But the evidence for that, this is the monophyletic clade of chemosymbiotic bivalves that were sequenced. And the evidence really rests on this single taxon here, Benthomodiolus lignicola, which is basal and is found on woodfall and on whalefall habitats. And there is actually not very good resolution in that tree for the other groups. Nevertheless, so it's a really good hypothesis and a really good start. And really, the research into this idea of evolutionary stepping stones was focused on 3 groups in detail. The mitelids, which I'm going to talk about first, the vesicomyids, and rather less amount of research, actually. And then my favourite taxon and the animals that I actually work on, which is cibberglinid annelids, and some other annelids as well, which we will have at the end if we have time. So that work on the mussels, Dan Distel work, really the most important update of that happened in 2006, the work out of Bob Reinhoek's lab and his colleagues. And this was their sort of summary of their evolutionary thinking in terms of what role habitat has played in driving evolution in this group. And they were quite circumspect in their discussion, actually, about how important whale falls and wood is in driving evolution. But certainly in their reconstructions of, here, let's look at this one, in reconstructions in the lower tree here. Oh yeah, I think it's complicated. Is that it is certainly most sort of parsimonious and most in terms of the maximum likelihood that the ancestral habitat state, even with this massively increased sample set, was wood or bone. You can see here the vent animals form a derived clade, and most of the diversity is in the seep, and then more basal forms with the wood and bone. But again, based on only one or two wood or bone taxa. Alors maintenant, c'est nécessaire parler en français. And I said to Marina that I would speak a little different language today because she was berating me for the fact that I able to speak in my native language. But this is a remarkable period that's been driven by the Paris Museum in particular, and I name it Recherche sur les moules. And we're going to just, oh, that's the right button. We're going to just kind of flick through a few of these papers. Another paper that looked and supporting this idea of Dan Distel et al., that the origin of these things was on wood and whale falls, and then several papers describing more species of mussels from vent, seeps, and organic fall environments. And I like to think that this may be a little bit driven by perhaps the culinary delights of France and their excitement with mussels, but let's see about that, maybe not. So I'm going to summarize that using the most up-to-date phylogeny, which was in Thubeau et al. In 2013. And you'll see here that the taxon sampling has increased dramatically in those 13 years. So this is now the complete list of taxa. Everything in green is an organic fall, so a wood fall or a whale fall on the seabed. And those in blue are cold seeps, and the red things are the hydrothermal vent taxa. And it's now really, this is quite strong evidence, really, that the basal form is most likely to have been a woodfall or a whalefall group. And that's quite interesting, and I think that it does still support, certainly from the phylogenetic evidence, that original hypothesis of 2000. And just out of interest, the IDAS from the Aberdeen region, Scotland, has, John sent some sequences to this group, sent some specimens to this group, and they did sequence that, and that's where that falls. So in some ways, that story from 1900 has now come back finally into the molecular sort of world. So that's the story for the mussels, and we'll just now review some of the other groups, and particularly interesting, firstly, to look at the vesicomyids. So what role have whalefalls played, these organic carcasses on the seabed, and in phylogeny and diversification of this group? And I don't think that this is anything like the same sort of case as for the mussels. And in fact, the reason for that, really, there are not really any strong evidence of whale-specific or woodfall-specific vesicomyids. These are really specialist on vents and seeps, and you can see here that the most likely scenario is that these things have diversified across a range of chemosynthetic habitats, mostly seeps, have moved into vent habitats, and the only ones which are present on whalefalls are also found in seep or vent habitats. So this is really a species which is using these environments as ecological stepping stones. So moving on to the annelids, and a group which I've been working on extensively in the last 5 or 10 years with my colleagues. Well, let's just look at a really important paper. Again, a paper which is not so old that you don't cite it all the time in every grant proposal that you write. Van Dover et al. In 2002. Very, very good paper, very interesting. But let's just look at what they wrote. And they wrote that modern vestimentiferan tubeworms diversified in cold seeps. A very bold statement, actually. It doesn't. There's no maybes or no caveats. There's no well, maybe they diversify. You know, that's normally what one writes if you're not totally sure. And the evidence for this was a phylogenetic tree right here, showing that the hydrothermal vestimentiferans are derived clades to some extent from the cold seep ones. Here's a lamellibrachia cold seep. Actually, a hydrothermal vent lamellibrachia, but that's another story for another day. So this is an interesting, quite bold thing, and you'd think that would be a hypothesis that would sort of stick around for a while, but no. The discovery of a remarkable new clade of Siboglinid annelids, specialist on whalefalls. And this is the animal which Greg Rouse named in 2004. Shana Gafredi, who did the molecular work, and Bob Ryan, who led the development of the whalefall research at Monterey Bay. Osedax means bone eating. This was an entirely new clade of animals, which they showed straight away by molecular analysis. And after we've done that, it becomes more obvious from the morphology. But when you first look at these things, nobody knows what they were. I mean, that took many years before we even got to the stage of thinking that they were annelids related to vestimentifera tube worms, but they certainly are. And in their phylogenetic analysis, these two species of bone-eating worms, these are animals which I haven't explained what they are, so I want to focus on the evolution, but they are animals which bore into whale bones and grow on them. And here you can see one. It looks like a little bit like a flower growing out the side of the bone. This is the plume, it gets oxygen and it pumps that oxygen down to a consortium of bacteria, symbiotic bacteria in the root tissue, which extract oil from the bone. So these guys, they don't need sulfur oxidizers, they just eat the oil directly. You know, they just like skip that step from the bacterial point of view. And why not? You don't need to wait for the sulfide bacteria to create it for you. So this was interesting because Osedax in this analysis turned out to be the sister taxon to the hydrothermal vent and Lamellibrachia vestimentiferans, which with what are called the frenulates, which is another whole story. These are another group of annelids which were described many years before the vent discoveries, what called the Pogonopherans for a while, the beard worms. We now know they're also part of the Siboglinid annelid radiation. And that was the story, is that probably these are derived from an ancestor which lived in these reduced muds. So these live in reduced muddy habitats. This is other polychaetes, your ragworms on your beach, everything else. These are animals living in reduced sulfidic muds. These are living at vents and seeps, and these guys are living at whale bones. Well, of course, what happens when you publish a paper in Science is everyone else starts publishing as well, and we got lucky and got involved with a rather fortuitous discovery of a new species of Osedax in very, very shallow water in Sweden with my colleague Thomas Dahlgren. A Japanese group started sinking whale carcasses off Japan and studying those, and they described a new species. And this is just, not to bring it up to date, there's more comes after this, but we very quickly, as we started adding taxa to our phylogenetic analysis, Thomas Dahlgren did this work and I did the morphological work. But we found that Osedax actually still coming out as most closely related to the vestimentiferan worms, not to the frenulate mud dwellers. But that relationship was not very well supported. There was no strong nodal support there. So the world carried on. More species were described, Roseus. Then several more species have been informally described, and then we did an update on the phylogeny that came out last year, including some new species from the Antarctic. It turns out the Antarctic is like a bit of a hotspot of whalefalls. They're all over the place down there, and there's going to be a lot more species described soon from there by my colleagues and others. So just to where we've got to now with this story, and just to show you the rapid pace of research, is that tree from Greg and Shana in 2004 with 2 species is now this entire clade here of 20, I keep losing track. At the moment, it's something like 26 species of Osedax. Soon, at the rate we're going to be more species of bone-eating worms than any of the other groups of Siboglinids at the rate we're working on them. And it's quite interesting. We're now starting to see strong relationships within the Osedax clade, another story. But let's just look at the relationship. They now come out, as you add more taxa and more genes to the analysis, they come out as a sister group to the frenulates, which derived from the vestimentiferans. So a fundamental change in the topology of the tree, which is quite interesting, and shows that we maybe have to rethink this a little bit. Now, at the same time, I want to briefly mention some of the work because it is relevant on the other groups. This includes the wonderful Chrysopatelid, what Helena Victland, who's here as well, my colleague working on these, called the carpet worms, the snowboarding worms. This is animals, so these are the scale worms, the polychaetes, which live on hydrothermal vents and on whale carcasses, and they're an example of a stepping stone species in ecological time. And then a rather remarkable species, there's not time to talk about it, but this is found on a whale carcass. It's actually most closely related to the methane hydrate ice worm, which was a famous worm when it was found. It was living in blocks of methane hydrate. And that's most close relative. So there's a close link between the whalebone fauna and potentially methane hydrate. Hasn't been followed up with more work on that, but that would be very interesting to do. But most particularly on a group called the Dorvaleids, which Helena and I are fond of calling the candy floss worms, because they eat this white bacterial mat and they spin it around and make little pellets. So we nicknamed them candy floss worms for that. And Helena and our colleagues, and we've been describing many species of these from a whole series of whalefall experiments around the world. And we have a slightly different pattern here, where this is the last paper that Helena published in 2012. And here you've actually got an animal which we know is specialist at eating these bacterial mats. And they've invaded, this is the clade that we're rapidly describing more and more species of. So maybe it's a sort of the equivalent of leucinids in terms of its... nobody wants to look at them because they're really hard to identify and they're really small, but they're really diverse and probably quite important. And they're everywhere, all chemosynthetic habitats, and certainly invaded whale and vent environments multiple times. There's no obvious phylogeographic pattern. But this little image here, this is their jaws. They use those to scrape the bacteria off and eat it. The shallow water relatives use it to scrape algae off the rocks. The deep sea ones at vents use it to scrape bacteria off the rock or the bone or whatever. And these things, I don't think that they care. If there's a bacterial mat, they'll go there. If it's a whalebone or a seep or a hydrothermal vent, whatever, they'll get there, they'll get married, have babies, their babies' babies will have babies, and eventually, after a few of that generations, they will become a separate species, and that's how it works. And that may be one of the reasons that there is so much speciation in this group that's not really related to habitat use. So let's try and sum up. Little cartoons that I'm making. You're welcome to trash this hypothesis totally, because it's going to be submitted fairly soon, in 3 weeks' time, in a review paper that Craig and I are writing. So you have the opportunity today to feed back into that. If I've ignored a major paper, you can email it to me and I will make sure I cite it. But here's one scenario, which is the wooden steps scenario, is that animals evolve organic falls, wood or whalebone. We're not entirely sure which from those, but they certainly are able to migrate to vent or seep habitats and diversify there. Meanwhile, also diversifying here on whale, bone and wood fall habitats, organic falls. And that's sort of, with quite a lot of caveats, what we kind of think best evidence from phylogenetic data for the mycelids. It may not be particularly well supported by the fossil data, but that's the best evidence at the moment from the phylogenetic reconstructions for mycelids. So then we've got an example where things radiate from a vent or seep ancestor into clades that specialize in seeps, clades in vents and seeps, and clades just in vents. And in a couple of scenarios, those animals may be able to hop across in ecological time onto whale carcasses. And I think this is what's going on with a group like the Vesicomyids. So that's really an ecological stepping stone. And then the third scenario, the third sort of hypothesis, is that these whale falls can actually act as hotspots of adaptive radiation. And this is probably a vent or a seep origin. Let's not forget that vents and seeps have been around for a lot longer than large whale carcasses, and probably longer than even large boned other marine creatures such as plesiosaurs. So they evolved there, diversified, maybe specialized, so clades of them on vents. This might be your Vestimentifrons on vents, this might be your frenulates. But somewhere along the line, some of these guys hopped onto whale carcasses and then proceeded to speciate at an extremely rapid rate. Whale falls being isolated islands all over the seabed, being extremely conducive to speciation. And I think this is the story for Sibiglinid annelids. So that's where we're at. And I'd like to thank you very much. I'll take any questions.
Thanks, Adrian. We've got time for a couple of questions.
The
Of the mitochondria, did you say? Well, that's an important gene to look at because it shows us a lot about the degree of differentiation at a species level. That's the cytochrome oxidase 1 gene. So that's the reason that we're looking at that gene. It's not because we're studying the mitochondria. It's just because we're looking at trying to look at anywhere where we can see differences between the organisms that inform us either about their which species they are or where they fit in an evolutionary tree of life.
How long did it take these annelids to find these whale carcasses? So the oldest whale, fossil whale, does it have evidence of Osedax?
Well, there's a little clue. One little clue is that they have, it's not very common, but vestimentifrans have been known to hop onto whale bones. There was Escarpia spicata, I think it was, described living on one of the whale bones. You don't see it very much. My colleagues in Japan have put a whale bone in a tank that has lamellibrachia, shallow water vestimentifran, and it grew all over the whalebone just like that. We don't see that in natural ecological scenarios, but that implies to me that those animals were able to pretty quickly jump across, in the space of, like weeks and months. So even if they're not inherently specialist on those habitats. When we drive around the seafloor, you don't find lamellibrachia growing on whale bones. You find Osedax and idas and these other things. But we know that they can hop across. So that implies it probably happened quite quickly, for me, in my view.
When you had like last cartoons you had with the sort of different evolutionary stories, if you compare the different groups that underwent these variations, do you see any ecological similarities or any teaching strategy similarities between these groups that share a certain evolutionary scenario?
Not sure I fully understand. I mean, they're all, with the exception of Osedax, they're all feeding chemosynthetically, except for some of the mitelids we don't know about. But
some like epifaunal, like the backmodalians, like epifaunal.
yeah, I haven't looked at that. I have for the mussels. I haven't looked at that. No. So for the annelids, I mean, yes. I mean, is the answer. They're strikingly different. So vestimentifrans living on hard substrate chimneys are totally different in terms of where they exist to something like sibaglinum, which is within sediments in relatively shallow water in marginal settings, or Osedax. In sibaglinum, some of those, I mean, they feed using chemoautotrophy, but some of it's methane-based in some species. But the Osedax, of course, have probably just ditched all of their chemosynthesis. So, like, what's the point of doing that? We're just going to get straight in and eat the bones. And that's an example where, I think what you're hinting at, which is a fundamental sort of trait, has allowed them to do something else quite different. So,
Have you examined ichthyosaur skeletons for evidence of
we've started. Many groups have started looking, and one of the ways of
Richard up here might have some insights.
there's been a couple of not papers published on, but we've published a few papers. Stefan has and we have on looking at the trace fossil that Osedax makes in fossil whale bones. So we know what to look for. Hasn't been found yet, unless anyone knows of unpublished data in plesiosaurs, ichthyosaurs. But that would be very interesting. If I have time, I mean, it's relevant. I mean, I didn't discuss the molecular clock data. Bob Reinhoek in his paper in 2009 looked at that, and using two calibration methods, you either have the evolution of Osedax, roughly late Eocene, coinciding with the evolution of large oceanic whales, or going back to sort of late Cretaceous, a huge, that's a big period in many millions of years. So that doesn't help us that much. The smoking gun will be a good Osedax trace fossil in an ichthyosaur. So yeah, just keep looking for everyone who's.
Well, there is one paper which has described small provanid gastropods which are obligated to chemosynthetic environments from two plesiosaur skeletons in Japan. So there are some interesting hints that Mesozoic marine reptiles would also support similar communities in the past.
And we do know from modern experimental studies that you can get Osedax to grow on cow bones, even fish bones that have been put down close to a whale carcass. So we know that they don't really care what kind of bone it is from a purely biochemical, ecological, chemical point of view. So it's unlike, that wouldn't be, prerequisite to eat bone, basically.
Just a very quick addition to this. I mean, with the fossil record of Osedax traces, which is pretty small as well, and so forth, what this tells us is that as soon as there were ocean-going waves who dropped down to the deep sea floor, there was Osedax colonizing. So that adaptation, if you like, we don't know when it originated, but it looks like, at least with the waves, they as soon as this food source was available, there was an organism evolved that to use it.
Yeah, it was pretty quick. Yeah. No, that's certainly what the genetic data would sort of hint at as well. So yeah, it's in green. Yes, that is a slight issue with that. Now, I mean, one theory, maybe it was they used fish bones, for a while to keep the going. Now, that's a slight problem with the Cretaceous Osedax hypothesis. Yeah.
Right, I think we need a break.
Oh, there we go. I've got my power thing. Yep, I'm running on reserve.
So we will reconvene at 4 o'clock.